Hello, and welcome to the exciting world of DNA and RNA. And so now we're going to get into the very, very basics on why you are who you are. We know that we get all of our genetics from our parents, and those genetics come together to make our own individual DNA, and that individual DNA is what makes us who we are. But there are a few things that go into this, and there's a lot of information in this video. I will be adding a bunch of notes out throughout every single slide, so make sure you're leaving room in between each slide. I'll try to be better at telling you where I'm going to be adding quite a bit so that you're not adding pages during class time and you're not getting mad at me for this. So DNA and RNA, basically the instruction manual for our body. And so how this happens, we will get into here in a second. After this video, you should be able to state the central dogma of molecular biology. You should be able to outline the discoveries that led to the knowledge of DNA structure and function. And then you should be able to describe the structure of RNA and identify the three main types of RNA. Pretty basic stuff. Or is it? Dun, dun, dun. All right, so here we go. What's the central dogma? All right, so before we even go on, the word dogma. Some of you have never heard of this word. Well, dogma, besides the fact that it's a 1999 movie, is the idea that there's a set of principles and that is the whole truth and the basic truth about one concept, whatever that concept may be. So it's the central belief. The central dogma in biology is the idea of DNA to RNA to protein. And my arrows didn't draw in, so I will add those. But basically what you're learning about is the very basic concept of molecular biology, or just biology in general. Like, how do we get the way we are? Like, we were obviously not this full-grown person when we were born. When we were conceived, we were one cell. So how did we get from one cell to a person who can walk and talk and write and listen to this video and think this is so dumb? What's central dogma mean? Yes, so the central dogma is the basic principles of whatever we're holding to be true or whatever we're learning. So the basic principle of molecular biology. So right now we know that DNA is found in chromosomes, and those chromosomes are found in our nucleus of every cell in our body. Now, the DNA always stays in the nucleus. So how is it that the DNA is the instruction manual to make everything in our body, but it doesn't go anywhere? How do we get that messaging or that instruction code out to the cell? And so the basic idea here, or the central dogma, like I've been saying, is that DNA makes RNA, which then goes into proteins. So basically DNA to RNA to proteins. That's all I want you to know right now. DNA to RNA to proteins. So let's take a little backtrack in the history of the DNA. Deoxyribonucleic acid, also known as DNA, is the genetic material in your cells. We will call it DNA just for the sake of tongue twisters. And so there was a series of serious and real and true and great labs that were done to help discover DNA and how it is the true DNA, or the true code of our body. Because believe it or not, it wasn't always known, or DNA wasn't always known to be the core component to what makes us who we are. So we have a few scientists that we're going to know. The first guy I want you to know is by the name of Frederick Griffith. And Frederick Griffith made the discovery that there is some kind of genetic material that helps create the organisms that we are. And I am going to be adding quite a bit to every single one of these. I'm going to describe the procedures or labs that they did, the experiments that these guys did. So please make sure you leave room in between. So Griffith searches for genetic material that can change characteristics of organisms. So basically, he starts off, or he creates this idea that there is a genetic material, that there is something that helps code for who we are. Now, another guy by the name of Oswald Avery questions Griffith's results. So in his experiment, he decides to think, okay, so there is a genetic material out there. What is that genetic material? And so he wants to discover what exactly that is. So he ends up making this huge discovery. He ends up discovering through his experiment that DNA is a substance that controls the characteristics of organisms. So pretty big stuff. We got Frederick Griffith who says there is a genetic material out there. And then you have Oswald Avery, who comes in and says it's DNA. So now, believe it or not everybody believes that DNA is the big honcho. They're like, nope, that's not true. So then a new guy comes in, or two new guys by the name of Alfred Hershey and Martha Chase. So I guess I said guy, you know what I mean. So Alfred Hershey and Martha Chase, they end up doing an experiment using viruses and bacteria that confirms that DNA is most def the genetic material that makes up every organism. So huge experiments done, and these things were done in like the 50s. Okay, so if people are doing that in the 50s, and now we are wasting our time on cell phones, we are doing something wrong. Right? We need to step up our game. All right, anyway, you are expected to know these experiments, so I will give you the details of all these experiments. I'm not going to make it long and boring. It's going to be like bullet points of the basics of what happened in every one of those experiments. And because it's really important to know how all of this leads into the idea that DNA is the genetic material that makes us who we are. DNA continued. Now, believe it or not, just knowing that DNA is the genetic material was just the tip of the iceberg. Now we have so many more questions like, well, what does it look like? What exactly is it doing? Why is it important? And so these are all questions that are slowly starting to be questioned, experimented, and run through. Now we have a guy by the name of Erwin Chargaff, and he's a biologist, molecular biologist, and he studies DNA from many different species, not just from humans, because, we're always so selfish, we think about ourselves, but he studies from a whole bunch of different species. And what he finds is that, he doesn't find this out, but he ends up finding this interesting, that the 4 nitrogen bases of DNA, because DNA is made up of 4 different bases, adenine, guanine, cytosine, and thymine, A, G, C, and T. And he finds that that 2 of the bases match really well with each other, while the other 2 match really well with each other. So he didn't think this was a coincidence. He ends up coming up with these rules known as Chargaff's rules. And what he says is that A binds with T and G binds with C. Basics of the rules. I mean, there's a little more to it, but that's really what I want you to know right now. And this becomes really important when we figure out the DNA structure. So in the DNA structure, it was discovered that the DNA looked like a double helix or like a staircase. So it was found to be the genetic material. They wanted to know more about it. So there's 2 guys by the name of James Watson and Francis Crick. Now, unfortunately, these 2 are actually not as great as you would think. They got a Nobel Prize for something they kind of stole. So basically, they published in a scientific journal that the DNA structure is actually a double helix. Now, unfortunately, that wasn't actually their discovery in the first place. They took some work from a woman by the name of Rosalind Franklin, and she found that she was the first to actually find the DNA structure using X-rays. Now, unfortunately, the Nobel Prize cannot be given to a person who died. And because of the fact that Rosalind Franklin did so many experiments with X-rays, she ended up getting cancer and dying before the Nobel Prize was given for the structure of DNA. So Watson and Crick are actually typically given the credit for discovering the structure of DNA when truly they weren't. That's my rant on that, and I'll probably get into it a few more times. Why not? But either way, the point is that based off of this double helical structure, we now understand how Chargaff's rules can apply better to this situation. And I'm not going to try to draw, but obviously I am. So the double helix structure, that does not look like a staircase at all. I'm not even going to lie. Just ignore that. I'll show you a picture in class because my gosh, that's embarrassing. Either way, we know that DNA is a nucleic acid and that it has 2 nucleotide chains. And what that means is that there's like 2 bands, and those twist around to make that staircase look. Now, each of these nucleotide chains is made up of an individual nucleotide. And all of those individual nucleotides have a sugar, a phosphate group, and a nitrogen-containing base, those A's, C's, G's, or T's. When they come together, they're bonded in a way so that the A's and the T's only touch and the C's and the G's only touch. That's really important. So make sure that we know that. And in class, I'm going to show you what I'm talking about with the actual model. Now the fun stuff. Why is it even important that we know about the structure of DNA? It does what it does. Why do we care? Well, it's important because it actually keeps you alive. Funny how that goes, right? So DNA, yeah, it just sits there, but we know that it replicates during the S phase of mitosis, right? Hopefully you remember this. So the S phase, DNA is replicating or copying itself. So basically the breakdown, and it's actually a lot more complicated than this, but I've broken it down in the most basic terms. DNA is broken in half by an enzyme. Another enzyme, so a very different enzyme, pairs the nucleotides with an appropriate pair. So that means if there's an A that's empty, it pairs it with a T. And if there's a C that's empty, it pairs it with a G. And then after all of this after that DNA strand is read, we then have the complete copy of the DNA, and then we have two identical daughter cells, which will have the same type of DNA. Basics. We are going to get in much more detail on that. Don't you worry. And then finally, the very last thing I want to talk about was what's known as RNA. I feel like I've talked super fast in this video, so I'm super sorry. I really don't know what else to say about that, except maybe I'm sorry. All right, so RNA. Now remember, DNA stays in the nucleus. It's pretty protected. It's like it likes its home. It doesn't want to leave. Very introverted. RNA, on the other hand, is not so introverted. RNA is actually a very big helper. Remember the central dogma. We have DNA to RNA to proteins. Proteins are our workers of our body. DNA is the instruction manual. So what the heck does RNA do? Well, because DNA lives in the nucleus and proteins are found outside of the nucleus, we need a runner. So we have what's known as RNA. And there are different types of RNA. So basically, RNA helps pass information to make proteins. It is also a nucleic acid, just like DNA, but it's different in many ways. I'm going to be giving you those ways in class, so don't worry about it now. But there are 3 different ways that they are different, and you will be expected to know that. And there are 3 different types of RNA. We have what's known as messenger RNA, or mRNA, ribosomal RNA, rRNA, and transfer RNA, or tRNA. 3 different types. And we're going to go into detail on what those are in class. So I know I talked about this really quickly. I'm almost out of breath. That's like, bleh. But I wanted to make sure I got all of this information in one video. We are going to go through this slow. This is what we'll spend the rest of the week on. So don't chip if you are super confused right now. We are even going to do a fun little project. Hopefully you enjoy it. If you have any questions, ask me on Edpuzzle, please.